introduce my colleague, Dr. Brian Jensen. So Brian is a physician scientist, leading expert in the field of molecular and translational heart failure research. And he's a current professor of pharmacology and professor of medicine at UNC, and also the director of the UNC Catecholamine Heart Institute. So our history goes back a little bit when we met in UCSF. So before that, a little bit background. Brian got his bachelor's degree from Pomona College and went to UNC for medical school training. Then he did the residency in Harvard Medical School and the Brigham and Women's Hospital. After that, he went to UCSF for his fellowship in cardiology. That's where we met together in UCSF, collaborated, and did a lot of translational research, which helped identify B1 reduction in heart failure patients. And so after his training, Brian went back to UNC Medical School, started his faculty position, and promoted there. As I mentioned before, he's currently the director of the McAllister Heart Institute Professor of Pharmacology and Medicine. And his lab focuses on using molecular and translational approaches to study heart failure and the molecular responses to myocardial injury. And the three major areas, as we will hear in one area, is alpha-1 adrenergic receptors. And he focuses on how alpha-1 adrenergic receptor regulates cardiomyocyte metabolism and how we can use alpha-1 agonist to treat heart failure. As outlined in his talk today, is the adaptive arm of the cardiac response to recorded catecholamine stimulation. And other aspect of his research laboratory also focus on mechanisms underlying cardiotoxicity of cancer therapies and identify cardiac protective roles of nuclear receptors in regulating metabolism and inflammation. He has published over 100 papers in high impact journals. I'm not going to get into the list. He's on many editorial board and study sections, and he really established his name as a leading expert in the field. Let's welcome Brian for his work.
Thank you. I think I'm going to switch over to this other slide. Okay, so thank you to both Robin and Ting for the incredibly kind and probably over generous introduction. Thank you also for the opportunity to come and speak with you. I have told multiple people with whom I've met this morning that I kind of spy on the work that you guys do here, because as far as I'm concerned, you are one of the two institutions in the country that do cellular through human-based work in a very thoroughgoing and convincing and informative way. And so I've been spying on some of you already, and it's a real pleasure to get a chance to meet with you in person. We are going to talk about, there we go. So we're going to talk about the area of focus in my lab that I've been working on the longest, and this dates, in fact, all the way back to the time that I was at UCSF. This is not, but does it advance? Okay, I can use the mouse. Yeah, no problem. So I've been studying alpha-1 adrenergic receptors for years, and this is my one disclosure. I am, as Ting mentioned, keenly interested in developing a drug that might target alpha-1s, and I hope by the end of the talk you might understand why I'm enthusiastic about this project. I want to start this talk like I start every talk by acknowledging the people who do the work in the lab, who inspire me with their ideas. This is my lab at our recent holiday gathering. I want to mention alumni and collaborators as well. I have an additional acknowledgement. which is that I have known these two for quite some time. This is a paper on which I served as a technician for them primarily. This has gone on to be one of the most highly cited papers on which I've participated. The acknowledgement here is for the friendship that Ting Ting has shared with me throughout the years, and to Robin, actually, who gave me some advice back then that really changed my career in a favorable way. And I think hearing the words that I heard from Robin, which I won't share with you today, put me on a path to succeed where I wasn't before. So I have a deep bit of gratitude and acknowledgement for Robin as well. So with that, this is what we do. Ting Ting told you a little bit about it. We're going to focus today on alpha-1 adrenergic receptors. So they're GPCRs, alpha-1s are coupled to GQ. This is going to be a pretty translational and actually overtly clinical talk. And so we are going to talk about some of these synthetic agonists that I call out up there, as well as the endogenous catecholamines, norepinephrine and epinephrine. From a physiological standpoint, when people think about alpha-1s, honestly, I think mostly about alpha-1s in vascular smooth muscle. That's where they've been most studied. It's where they're clinically actionable at this point in time. I'm going to try to make the case to you that the alpha-1s that are present in the myocardium actually do physiologically important things as well, and that in the context of heart failure, which is the context in which I spend most of my time, that they do good things as opposed to the alpha-1s in vascular smooth muscle. So speaking of the context of heart failure, these are 40-year-old data demonstrating that in human beings with heart failure, that survival is inversely proportional to the level of circulating norepinephrine. There are other data sets that look similar to this, but these data and others have basically informed a paradigm that has existed for decades, suggests that the chronic catecholaminergic surge associated with heart failure is uniformly detrimental. And I'm going to try and push back a little bit against that paradigm today. I'm going to start with human loss of function in a historical context. The branding people at UNC have asked us to use these slides now, and I didn't think I would like it. It turns out I don't like it, so this is the last time I'm going to use this. So it's just silly. So the observational data regarding circulating norepinephrine were bolstered by the well-established fact that when you use beta blockers, drugs that block beta adrenergic receptors, in the context of heart failure, people live longer. This is indisputable. It's a class 1 indication, right? And so this has further informed this paradigm that suggests that catecholaminergic surge is uniformly detrimental. From an alpha-1 human loss of function standpoint, the first data point that we got was from the ALLHAT trial, which was published right at the end of my, right at the start of my residency, actually. At the time, it's the largest antihypertensive trial ever published. It's 24,000 people enrolled. There were multiple different arms. I won't bore you with details. What I want to call your attention is that one of the arms was doxazosin, which was an antagonist of all three alpha-1 subtypes. They've introduced new alpha-1 subtypes today. At the time, this was one of the first-line agents for treating blood pressure in humans. The Data Safety Monitoring Board stopped that arm of the trial prematurely because there was a twofold increase in incident heart failure in the people who got doxazosin. Primarily, the consequence of that was that doxazosin is now a third-line agent for treating high blood pressure. There was not a lot more thought given to the heart failure signal beyond that, except from a few people, including my mentor, Paul Simpson. So somewhat more recently, there has been a look at sort of a population level at patients taking alpha blockers. And now these days, alpha blockers, drugs that block alpha-1 adrenergic receptors, are used principally for treating benign prostatic hyperplasia, a very common problem in men. This study published out of Canada also showed that there was a risk signal for incident heart failure in patients who got alpha blockers. But they're really, boy, that reproduced poorly. You're going to have to take my word for it. That this is not so long. Actually, there are other studies, and this is one that I particularly want to call out. This is Jack heart failure paper suggesting that use of alpha blockers is not detrimental. In fact, this is a heart failure patient population from the VA, and patients with heart failure, diagnosed heart failure at the VA who got alpha blockers did every bit as well as those who didn't, and maybe even a little bit better. So I think there's still some equipoise here in the field. And this is going to be, we're going to talk more about that equipoise towards the latter end of this talk. There's that silly argyle again. So let's talk about mouse loss of function. And here I want to call out something really important. We are focusing on, I've talked here before about alpha-1s most broadly speaking. We're focusing on the alpha-1A subtype. There are 3 different subtypes. I'm going to talk very little about beta receptors, which certainly are more abundant than alpha receptors in myocardium. But when you hear me say alpha-1s, from here to far, I'm really going to, going forward, I'm really going to be focusing on the alpha-1A subtype. And I'm going to detour briefly because I want to show some data. This is the first time I've showed these data. They're unpublished. So this presentation might be a bit rough, but this is one of these sort of scientific serendipity stories. As Tinky mentioned, we're interested in a nuclear receptor called ROR alpha. And as a part of that, in collaboration with Anton Jensen over at NIHS, they did this is long enough ago, it was an RNA microarray on hearts of mice that have globally lack functional ROR alpha. And here are the top 7 genes that were ranked by differential abundance, decrease in abundance in the knockout animals, if you will, versus the wild type. And by pure chance, our gene, Adrenergic, which encodes the alpha-1A adrenergic receptor, was ranked 6th. So this is just pure serendipity, right? So we were interested. So the ROR alpha functions as a transcription factor. And we're interested in part because there's actually nothing known about transcriptional regulation of alpha-1A abundance, which is surprising, but I cannot find anything in the literature. And so it would be interesting if we had identified a putative regulator of alpha-1A transcription. So this is work led by one of my postdocs, Raghu. We first did some qPCR both in this, the SGSG is the global null animal, they're called stagger animals, and I won't give you their backstory, but we didn't find quite the downregulation in our hands as the microarray did, but certainly it was recapitulated. And then we generated a cardiomyocyte specific ROR alpha knockout and found that the same was true there, that there was considerably less alpha-1A within those hearts. We used both pharmacologic and genetic gain and loss of function here to show that the activity of ROR alpha was directly proportional to, or rather, the abundance of Adrenergic was directly proportional to the ROR alpha activity. This is using some established agonists and antagonists, as well as a constitutive overexpressor and some shRNA. So we were encouraged by that. This is all still correlative, not directly proving. But so we went on to ask whether we could prove that ROR alpha might directly regulate Adrenergic transcription. So the ROR alpha binds to a consensus sequence that is collectively referred to as RORE or E or EBOS. And here you'll see, and I think the pointer is just not going to work, but hopefully you can see at the top, there's E1 and E2. So there are 2 ROREs of EBOS, the promoter of Adrenergic. We mutated the upstream one, E2, and found using the luciferase assay that there was a clear decrease in alpha-1A expression as measured by luciferase activity when you mutated that site. So we thought that was encouraging to be sure. Then we did some chip PCR and found that when you pull down with an RR alpha antibody, that there was a clear increase in the abundance of Adrenergic transcripts, and that abundance was actually enhanced in the setting of hypoxia. there's more about hypoxia in this manuscript, but I won't get into it now. So anyway, we thought that fairly convincingly demonstrated that we had identified what I think is the first direct regulator of alpha-1A transcription. So back to actually, there's one more thing on this. This we might move forward with. So I don't know if any of you are familiar with CircuitDB. So that it's this online database of looking at oscillation, circadian oscillation of transcript abundance in various tissues, and they do many, not all genes. It turns out that the only adrenergic receptor that oscillates in the heart is our receptor, the alpha-1A. That was interesting to me in and of itself. ROR alpha is a known transcriptional regulator of circadian biology, and so we asked whether there might be some evidence that it might regulate the oscillatory expression of Adrenergic in the heart. This is difficult to see for you, I would guess, but it turns out that in our hands, the Adrenergic and ROR alpha oscillate at the same frequency. That BMAL, another known regulator of circadian rhythm, oscillates with the exact opposite frequency, which is known with respect to ROR alpha, that is. And that MYH6, which encodes alpha myosin heavy chain, didn't oscillate at all in our hands. This does not prove the direct regulation of alpha-1A oscillation, but we're super interested in this, and we're going to move forward with this, particularly in the context of regulation of circadian metabolism. Okay, now back to what we're actually here to talk about. So alpha-1As and alpha-1Bs are present on cardiomyocytes. We're really going to ignore the fact that alpha-1Ds are on coronary arteries for now. And I want to just say now and then leave behind the fact that there are no alpha-1s on cardiac fibroblasts. So this is work that was done before I joined Paul Simpson's lab. Paul was my mentor then and continues to be a mentor and collaborator. He and Tim O'Connell, who's now at the University of Minnesota, created a global knockout of the alpha-1A and alpha-1B. So any sympathetic signaling that occurred in cardiomyocytes in these animals occurred through beta receptors. These mice had small hearts and impaired effort tolerance. And I think more relevant to the work we're going to talk about going forward, they either died within a week after attack or developed exaggerated heart failure as evidenced by capillary hypertrophy and increased fibrosis. So one might conclude from that alpha-1 activation mitigates the toxicity of beta adrenergic receptor activation in heart failure. So this is sort of the point where I became engaged in this work. And when I started my own independent research program and really fits and starts super slowly at UNC, these were the 4 questions that I identified as most interesting to me. And we're going to try and walk through my attempts to answer them through the remainder of the talk. So let's start with the mechanisms question. So this is a sort of a look forward. We became interested in mitochondrial function in part on some surprising findings from a paper that we published in 2017. The purpose of this paper was to demonstrate that an alpha-1A agonist could protect against anthracycline-induced injury. We did some RNA-seq and found to our surprise that there was a really strong signal that in the animals that received the alpha-1A agonist, that they had preservation of transcripts in multiple different mitochondrial related pathways. And this was all new. There was no notion that alpha-1s might regulate mitochondrial abundance or function. Little bit of in vitro work. This was not a deep dive into mitochondrial function at all. Suggested that perhaps the alpha-1A agonist might be objective for preserving mitochondrial membrane retention. So that prompted us to launch into this work, which was led by a grad student, Peyton Sandroni, published, I can't even say last year anymore, year before last, I guess, where we used a global alpha-1A knockout mouse to ask whether alpha-1As might participate in the mitochondrial physiology of the heart. And I will start by saying that I did not expect that there would be a basal phenotype. In general, when you knock out adrenergic receptors in the heart, mice look pretty normal, right? It really takes some form of stimulus or insult to bring out any differences. So I was surprised to see that there were genuine morphologic differences in the mitochondria in the global alpha-1A knockout animals. We went on to collaborate. This is our first collaboration. We have ongoing collaborations with Kelsey Fisher-Wellman, who's now at Wake Forest, who is an absolutely brilliant mitochondrial physiologist. He's incredible. He also has sleeves of tattoos on both arms, which is awesome. So he has developed a system that he's called mitochondrial diagnostics, and here I'm showing you some respirometry that he uses, titrating up through physiological phosphocreatine concentrations to show that when supported by fatty acids, which are the primary substrate for oxidative metabolism of cardiomyocytes, that the global 1A knockouts, both in permeabilized muscle fibers and in isolated mitochondria, perform worse, have less oxidative capacity than wild types. We wanted to understand as best we could why that might be the case, and the fatty acid signal led us to look at a couple of things. So we did some targeted lipidomics and found that there was appeared to be a block in the metabolism of long chain isocarnitines in the heart. And so you can see here that in the knockout animals, they're more abundant than the wild types. This is not to be confused with actual flux work, but this was a signal at least. And so we went on to look at the various steps in beta fatty acid oxidation and identified that LDAD, long chain dehydrogenase activity was lower in the knockout animals than in the wild types. We and Kelsey both looked at the first 4 enzymes in the electron transport system and found that 3 of them had lower activity in the knockouts than the wild types. So we felt that this offered some evidence that animals, at least animals globally lacked alpha-1As, had impaired mitochondrial function that was related to fatty acid supported oxidative phosphorylation.
Right. I have a quick question.
Sure.
You're probably going to touch on this.
Yeah.
I was just thinking about the alpha-1A. It's actually very interesting. But the quick question I have is whether they are redistributed, because you show in the memory, right? Where are they? What's the time?
So this is a great question. This is not work that I can take any credit for, but it turns out that at least some of the important physiologic functions of alpha-1As require nuclear localization. So they go to the nuclear membrane. This is Tim O'Connell's work, which is really, really interesting.
Right.
And not anything that we've explored in our work.
Sure.
Yeah. So we next moved on to this question, right? Whether the adverse phenotype of the systemic double knockouts that Paul and Tim created was mediated by alpha-1s in the heart. And so we created a cardiomyocyte-specific knockout mouse, and this was work led by J.D. Jung, who's sadly left science, and he's an interventional cardiologist for Stavros. We were talking about this earlier. He's stenting people and making a bunch of money, even though he was a great scientist. So we made this mouse. And as I said before, the phenotype, the cardiac phenotype, the basal phenotype is indistinguishable from wild type rare base. But when you subjected them to a permanent left coronary artery ligation, they fell apart. You can see they died quickly. And those that died, most of them had cardiac rupture, the particular three-wall rupture. It was a really striking phenotype, both the death and the three-wall rupture. JD was super interested in this three-wall rupture, so he he tracked down these folks at North Carolina State. So our BME group is split between UNC and NC State, and they're really great. These folks happen to be in Raleigh. They had this apparatus called a Biotester 5000 biaxial test. I think it sounds like something out of sci-fi. I just love it. It is an apparatus that is able to test the tensile properties of tissues. And so he, rightly or wrongly, took some infarcted mouse hearts over there and mounted them on this apparatus. We first wanted to ask whether the stiffness of infarcted heart was different from sham-operated animals, and it clearly appears to be the case. And then we asked whether there would be a genotype-driven difference, right? And so here we're just showing the knockout animals versus the wild type. And here, clearly, you can see that there is a difference. The Young's modulus is different in the knockouts than the wild types. Yes.
Can I ask a question?
Of course.
So how soon was the heart isolated for this analysis?
Three days after.
So the increase in stiffness happened very quickly.
Yes, for sure. And we used 3 days because so many of them died. Like 5 days, we had too much attrition. So this is a relatively early time point. And all of the data I show you going forward are day 3 time points. We had anticipated going in using day 7 or day 14 time points. Too many mice died. So we did some cardiac MRI and found that the contractional function was worse in the knockouts than the wild types. The infant area was larger in the knockouts than the wild types. And, as a biomarker, we chose HMGB1 rather than troponin, which is used clinically, in part because the mouse troponin assays are not great, and in part because we were interested in necrosis in particular. And I'll tell you in a minute why we were interested in necrosis. So we did, as one does, some RNA-seq, and here we tried our very best to sample tissue from the border zone or an analogous area in sham-operated animals. And then looking at this didn't read this well either, but looking at twofold upregulated transcripts differentially expressed in the knockout ligated animals versus the wild type ligated animals. We found one pathway enrichment that seemed to harmonize well with our finding, which is extracellular matrix structural constituent conferring tensile strength. That seemed to agree fairly well with what we've seen phenotypically. We also found pattern recognition receptor activity, which at the time were all words that I recognized independently, but when you put them together, I wasn't entirely sure what that meant. So pattern recognition, as I'm sure most everyone else in the audience knows. This finding led us to look at necroptosis, necroptosis being one of the many forms of programmed cell death mediated by PLR. And the 3 canonical regulators of necroptosis are RIP1, RIP3, and MLK. So we went on to do some Western blotting here for RIP1 and RIP3 and found that they were more abundantly expressed in the knockout than the wild type ligated animals. And then for MLKL, showing now sham operated animals just to demonstrate that necroptosis is inherently upregulated by ligation, but even more so in the animals that lacked alpha-1A and cardiomyocytes. We wanted to localize this findings of cardiomyocytes, so we did some immunofluorescence, and reassuringly to me, it certainly appeared that the MLKL signal was most abundant in myocytes rather than non-myocytes. As to further test this hypothesis that alpha-1As might be a brake on necroptosis, or rather than the loss of alpha-1As enhanced necroptosis, we used NEC1S, which is a necroptosis inhibitor, and that completely abrogated the difference between the two genotypes after ligation. So there's a summary. I don't want to pretend. So when you write a manuscript, you need to pretend that there's like a single mechanism that accounts for the finding that you're looking for. And that's what reviewers want to see, that one mechanism. I don't want to pretend that this is the only mechanism at play here. But I think we've presented enough evidence to suggest that this is one mechanism that's at play, at least. And how it's related to the mitochondrial findings, I think, is a very fair question and one that we proceed. So now we're going to pivot towards the clinical part of the talk. I told someone earlier today that though I'm a clinician, I've been a clinician for many years. I've always been scared of clinical research, and I've never really done it other than in a collaborative fashion until the past couple of years. But I am sufficiently motivated by this question that I was willing to do some clinical research. And so here we go. So these days, as I told you earlier, alpha-1 blockers are principally selected for the alpha-1A subtype, which is the subtype of interest for us, and B, used almost exclusively for the treatment of lower urinary tract symptoms related to benign prostatic hyperplasia. And they are used a lot. So tamsulosin, which is the flagship drug in this class, there are almost 6 million men in the country on this drug right now. It is a huge, huge drug. By and large, it is well tolerated. So we wanted to ask whether our mouse findings had clinical relevance, and so we first did a single center study. This was also led by J.D. John. We looked at patients who came into UNC with an MI, who got a standard PCI. And then we stratified them by whether or not they were taking alpha blockers and followed them out over a couple of years. We adjusted for age, sex, hypertension, and heart failure, so limited number of variables, right? Obviously, we did not adjust for everything that could possibly have influenced this outcome, but we found that there was a hazard ratio for death of 1.5 that was taking alpha blockers versus those that weren't. We were interested in testing this hypothesis in a larger data set, and so we went across the script to the UNC School of Public Health, which is the incredible School of Public Health. We're so fortunate to have them so close at hand. Here we worked with Chase Latour and Shola Roy, who helped us access this Medicare fee-for-service database sample. So 160,000 people here now. We used here as a comparator, a control group, we used people who are taking the second most commonly used class of drugs for BPH, which is 5-hour reductase inhibitors, just in hopes to try and equalize variables between the groups as much as we could. And then we used IPTW. By we, I mean the School of Public Health used IPTW to make sure that we could eliminate as many differences as we could across the 41 clinical and demographic variables that we measured. So we did that within the limits of this approach.
Hi, Brian. Good question.
Yeah.
I know this is already published.
Yeah.
I also read that, but I apologize. So in the context of the alpha-1A blockers,
Yeah.
So the patients that you recruited, do you pay attention to when they take the drug?
No.
Oh.
And so, I should say that none of these are all observation, retrospective. We haven't, I'm super interested in actually doing a prospective study. But
Sure.
no, so there's no, I hear what you're saying. It's a great question that I had thought about before, but no.
Okay.
So these were our pre-specified endpoints: heart failure, major adverse cardiac events, and mortality. In contrast to the LHAT findings, we actually found no difference in heart failure. But we did find a statistically significant, albeit small, difference in mortality MACE, the combined endpoint of heart failure MACE. And this is after one year of follow-up. So, caveat, important caveat. We adjusted for 41 different things, but it's certainly possible that there are some unmeasured variables that could have affected our results. So if you calculate the number needed to harm, which is derived from the risk increase that I showed you in the last slide, incur mortality, if you believe these statistics, and you believe our study, you need to prescribe an alpha blocker to 260 people. But when you think about the fact that there are 5.8 million men in the country taking this drug, that ends up being a pretty big number, and particularly we only examined outcomes of 1 year. So I didn't want this to be the last word on the topic because it shouldn't be. It's not a flawless study by any means. So we, yes.
Seems to me that from the data, the curve is diverging really negative.
I'm going to show you. Yeah. So it's
Just at least 1 year is pretty short.
I'm going to show you some data in a minute.
Instead of looking for
Yeah.
bigger number,
Yeah.
looking for longer.
Yeah. So I agree with you. And so I'm going to show you some unpublished data next that we moved on to examine this Trinetics database. So it's a huge online clinical database. Many millions of people. This was led by Logan Kirkland. That's not Logan Kirkland. He refused to share a picture with me, so I went on the internet and found somebody named Logan Kirkland, but that's who that is. Logan Kirkland is a postdoc in my lab who's applying for MD PhD programs this year. And it is not hyperbole to say he is probably the best scientist in the lab, including me. So if you're looking for a really good MSTP student for next year, Logan Kirkland, but not that Logan Kirkland. So we were interested in looking at another data set and looking at longer time points, longer follow-up. We started with 158 million people in the Trinetics. And after applying all of our inclusion criteria and doing some propensity score matching, we ended up with between 30 and 50,000 people per group here. Boy, that reproduced terribly. Hopefully, so the end point on the x-axis here is 5 years. And to your point, the curves do hew pretty closely together the first year or so, but then they really start to diverge. And so the hazard ratios that I'm presenting here are the 5-year hazard ratios for each of these endpoints. So it actually statistically significant hazard increase in hazard for stroke as well. Logan also compared selective alpha blockers versus non-selective alpha blockers, and here the differences were smaller, but still significant, showing a hazard for selective alpha 1A blockade. Where he has promised me that he has a near final version of this manuscript that I'll be looking at this weekend or early next week. So hopefully this will be out for public consumption soon. But hopefully does that address the question? Yeah, I agree with you. Okay, so gain of function. As I said, I am keenly interested in targeting alpha 1s with an agonist. So let me show you some data that hopefully will substantiate why. Here are the summary of the studies using varying degrees of sort of physiologic and grossly superphysiologic overexpression of alpha 1As in mouse models. Granting all of the shortcomings of overexpression of models, they all appear to be protective against various forms of insulin. We have used a couple of different alpha 1A agonists. This is dalusalgron. This is a drug that actually was in humans, as in the early 2000s, Roche developed this as a treatment for urinary incontinence. It was well tolerated. It's been in thousands of women, but they shelved it because it didn't work for urinary incontinence. So we had a bunch of this synthesized in Hong Kong, and we used it initially in an anthracycline-induced cardiotoxicity model. Eric using that really high dose that I told you about before. In any event, it first of all didn't increase blood pressure, which we thought was important because if you're going to target an alpha 1 receptor, as I mentioned up front, in the setting of heart failure, you really don't want to increase vascular tone. It's going to be detrimental. So the first question you have to ask with any of these drugs is does it affect blood pressure? And this one did not in our hands. It did protect against the decrement in contractile function induced by doxorubicin, and that protection was lost in alpha-1A knockout animals, suggesting that the protective effects do require the alpha-1A. We treated a lot more alpha-1A animals with doxorubicin than what you see there, but they all died. So hopefully reinforcing the point that I made earlier. So this is work that Paul Simpson's done. Paul has continued to work in this field until very recently. This is A61603. It's highly selective. It's highly potent. It's a tool compound. It's short acting. It's not a useful drug translationally, but it does make a good point, I hope. So TAC, this is a rescue experiment. So pumps infusing A6 were implanted 2 weeks after TAC. Hopefully you can see the trajectory of fractional shortening in the A6 treated animals trends upward in a very clear way, whereas the vehicle treated animals continue their downward trend in fractional shortening. He also used this drug in the, admittedly problematic or at least finicky high-fat diet LNA model of HFpEF, and found that A6 appeared to be protective with respect to diastolic function in the heart in this context. So these are all unpublished data that Paul has agreed to allow me to show. A6, this tool compound that I mentioned, also enhances mitochondrial function. You'll recall maybe that I showed earlier the loss of function side of this coin, wherein isolated mitochondria from global knockout alpha-1A animals have decreased oxidative capacity. Here we're comparing vehicle control treated animals with animals that are pumped with A6. The experiments that were done on the top were done in Kelsey Fisher-Blumman's lab. And A6 certainly appears to increase fatty acid supported oxidative capacity through the physiological spectrum of phosphocreatine. And that was true for both medium chain and long chain fatty acids. We talked earlier about LCAD, one of the enzymes, one of the free grain enzymes in the fatty acid oxidation spiral. I showed you that the absence of alpha-1A decreased LCAD activity. In contrast, administering A6 increases LCAD activity and also increases the activity of mitochondrial trifunctional protein, another one of the three main players in the beta fatty acid oxidation spiral. It did appear to increase the activity of complex 1 and complex 4 in ETS. And so collectively, we thought that this offered a nice gain of function counterpoint to the loss of function data. And these were published in our JNCC paper a couple of years ago. So I'm going to close with hopefully what I've showed you so far is relatively data supported. What I'm going to show you now is almost totally speculative. I'm going to show you some very early data that may or may not pan out. So, like lots of people these days, we're interested in how systemic metabolism interacts with cardiovascular health, inspired by the successes of SGLT1 and the GLP-1 drugs. So we went to the literature looking for a link between our receptor and systemic metabolism. We found this paper published 15 years ago in the Journal of Hypertension. So this is a relatively small sample size, 1,400 people from this Pamela cohort who had metabolic syndrome. And this was not a proper GWAS. This was a sort of a targeted look at 1,324 SNPs and 98 candidate genes that were selected based on the a priori possibility that they might regulate any of these processes, so like nervous system, oxidative stress, RAS and sodium balances. Of those, the SNP near our gene showed the strongest correlation with metabolic syndrome, with an odds ratio of 1.7, an adjusted p-value that seemed believable. This has not been, there's one other smaller study that showed something similar, but this has not been replicated in the larger data set. Nevertheless, it was interesting for us. So, like I said, speculative. This is just one cohort of animals. But we treated black 6 male mice with a high-fat diet. This is very high-fat diet, 6 kilocalorie high-fat diet. And then at week 6, we pumped them with either vehicle or this A61603 agonist. And those who got the vehicle, after an initial post-surgical dip showed a continued increase in weight. Those who got our drug did not. And similarly, the fat mass, this is by echo MRI, continued to trend upward in the vehicle-treated animals, but trended downward in the ACE-treated animals. We looked at heart rate, heart weight, and white adipose tissue weight, both of which were lower, white adipose tissue considerably lower. This is pooled from multiple tissue depots. And collectively, this suggested that A6 protected against the increasing adiposity associated with a high-fat diet. This is my last slide of any substance. It's super busy, and I don't expect you to read it. But the gist of it here is that, first of all, we hope that we have provided some evidence to suggest that maybe the paradigm wherein chronic catecholamine surge in heart failure is inherently detrimental might be incomplete. And that in fact, alpha-1s might offer a break on the detrimental effects of chronic adrenergic receptor stimulation. And then JD, who was the first author on this review, goes on to suggest that perhaps the best way to treat heart failure might be what he called comprehensive sympathetic modulation through the use of a beta blocker combined with an alpha-1A adrenergic receptor agonist. And we are keenly interested in pursuing the latter part of that question. So here are my take-home points. I'll read them to you. But hopefully I've provided enough data to support each of them. And again, I am really honored by the invitation to come and speak with you and meet with you individually. I really admire the work that you guys do here, and I'm happy to answer any questions that people have.
Robbie. Brian, this is really very exciting, intellectually challenging. It's very exciting. But I'm going to go back to the, I think you touched upon it, just, one of your last slides, with this relationship between beta and receptors, essentially. Maybe it's a good idea to target all the receptors, but then, the challenge then becomes all these off-target side effects and things. So, and then it takes me essentially to the circadian cycle complexity of this. When the patients take the drugs, could you specifically target alpha-1A in the morning, and beta in the evening, sort of thing? I don't know. Yeah. It's just a process here.
Yeah, no, I would, to take that a step further, when I saw the circadian oscillatory data, my first thought was, oh my God, have we missed the boat with all of the work that we've been doing before, not paying attention to the timing at which we acquire samples and everything. So we have to understand this better. Whether or not it has implications for human health, I don't know, but certainly for the work that we do, we need to pay closer attention to this. So we're, Roger, the postdoc who I showed you, who led that Morale project, is leading our effort to understand whether alpha-1As might regulate circadian metabolism. There's, go ahead, Rob.
No, I don't think that. But I just have thought, and I have a specific question to follow. But I don't know what I had mentioned in your humble and straightforward now as you were 60, 20 years ago. And you don't emphasize, I will do it for you, what a incredibly large task that you have ahead of you in going after the dogma of heart failure benefits from adrenergic blockade. And well, maybe adrenergic alpha adrenergic, but adrenergic agonist may actually help heart failure. And that, surely still is an uphill battle. And your approach, from a diversity of approaches, from the mouse model to going after the human data sets, which is already quite remarkable and consistent. One thing I missed is, how was A6 developed? Sorry, A616.
Yeah. So that was an Abbott drug.
So, and how did it come about, and how specific is it? Because it gives you an incredible tool to go after metabolism now.
Yeah.
And so, do you mind just going into a little bit of the background of that drug?
Sure.
And because the world that it opens up.
Yeah.
For these studies. Yeah.
For sure. I mentioned that this dabuzalbon compound that we've also used was developed by Roche in an effort to treat urinary incontinence. There was an effort across multiple different pharma companies in the late 90s to target alpha 1As for treatment of urinary incontinence. And so each of them tried it out on their series of compounds. A61603 was one of the Abbott compounds. Ended up being shelved because it's too short acting. It's just not tractable and usable clinically. But it is incredibly potent how it is selective for the alpha 1A over the other subtypes. Just for an example, but we use 10 nanomolar concentrations in vitro of A6. Wow. When Paul pumped his tack mice, he uses 10 nanograms per kilogram per day. So this is a super potent compound. But yeah, it was all there's a whole MedChem paper about the 35 different compounds that various companies developed to target alpha 1As for urinary incontinence. None of which made it to clinic.
And is there a hypertensive effect as well? Or
no, not in dabuzalbon. There's no effect on blood pressure. Yeah.
Really?
Yeah. Yeah. It's sure there would be if you gave it up, but I'm sure.
Yeah.
But within the right dose range, you can protect the heart without activating vascular smooth muscle. As far as we can tell.
Really?
Yeah.
Go ahead, please. Brian.
That's great. Thank you. You mentioned, I like this slide a lot. Blocking part of that inertial activation and activating the other. Robin talked about the dogma that we step back. This is the new dogma. The old dogma was blocking the adrenergic pathway.
Yeah.
Right?
Yeah.
That's how that was a crazy place for 30 years.
Yeah.
Right?
Yeah.
And now we are blocking the adrenergic pathway, but if you step back, this is how the body reacts. This is the hormones of flight and fight. So there is on the body. And we never really figured out how beta blockers really work. I mean, Mike Bristow just passed away. As you know, they gave him a lot of hard time at the beginning to reverse this dogma. So this idea that yin yang and maybe there's so are you looking at it as an isolated way to do it, or as in the figure from JD as a combo way? Block one activate the other one.
Yeah.
And then, or because we have medications that they are using, ETH and other things, and some of these people got beta block.
Yeah.
Although all this help, some people did not get beta block.
Yeah.
I'm sure there's going to be these subgroups in these studies.
Yeah.
Is this a combined kind of yang, or is it? I'm sure you follow that.
I have for sure. And to touch on the first observation you made, I actually consider the use of beta blockers in humans with heart failure to be one of the most courageous stories of translational science that I know of. But to go completely against dogma and give a drug that is a negative inotrope to patients with heart failure, like that is brave, and it worked. And we give all of our heart failure patients beta blockers now. So absolutely, we should continue to do that. And I want to talk about carvedilol at the end. So 100% should continue to do that. I think that if I could give you the true believers boiled down take, is that the chronic sympathetic surge clearly does bad things, or at least it's associated with bad things. There have been studies, Moxie and Moxcon, where they knocked out all sympathetic activation. Heart failure patients did worse. The best trial, heart failure patients did worse. So I do think that there's something to be said that you cannot, as you suggested, cannot disable the fight or flight response altogether in heart failure. And my true believers take is that that's partly because you lose the transduction of beneficial effects through the alpha-1 anchor if you knock down the sympathetic nervous system entirely. So yeah, I think, pie in the sky, beta blocker, alpha-1 agonist. Let me quickly talk about carvedilol because it's a question I get frequently. Right. So carvedilol is a beta blocker that also has alpha-1 blocking properties, and it is one of the 3 first-line beta blocking agents that we use with any regularity here in the States. I have periodically gotten questions, well, if blocking down alpha-1s is so bad, why is carvedilol so good for people? But it turns out that in some really nice human physiology studies, that the alpha blocking properties of carvedilol are extinguished after about a week or 2. That in chronic use, there is no activity of carvedilol at alpha-1s at all. Carvedilol benefit probably comes from beta blockade as well as biased signaling through the beta receptor that the Lefkowitz lab has illustrated really eloquently. It has some pleiotropic positive metabolic effects, but it's not the alpha-1 that accounts for carvedilol. What other questions?
That was fine.